this a little bit. Compare the different types of cell division. And so binary fission, that's the one that's carried out by prokaryotes. It's very simple. It doesn't have a lot of stages to it. It's basically the cell copies its DNA chromosome, it enlarges, and then it will split and divide into two. We have cytokinesis here as the final step. And in the end, we have two cells that are identical to the cell that produced it. And the original cell is no longer. It's basically has divided its contents into the two new cells. And mitosis and meiosis are found in eukaryotes. Mitosis has one set of cell division where we have a diploid cell resulting in two diploid cells. This is what all of your body cell, all eukaryote body cells do. They undergo mitosis when they need more of the cells or to replace or replenish. So it starts with a division of the nucleus going through the different stages of mitosis, which we'll go into in a minute, and then cytokinesis, and then we have two cells. Whereas meiosis, you can see there's two sets of divisions that happen. And we have a diploid cell that is undergoes, divides up its chromosomes. So you can see that the number of chromosomes after the first division is already half of what the original cell had. And so it comes down to a haploid number. That's why there's the N there. And then there's a second division where each of the chromatids actually separates and makes their way. So in the end, with meiosis, we have 4 cells that result, and they're haploid. And they're not identical to the cell that produced it. They actually have some of the genetic material, but not all of the genetic material. So there's 2 cytokinesis events, 2 dividing of the cells. So compare the phases of mitosis and meiosis. In mitosis, you know that the chromosomes get duplicated. They get duplicated actually prior to it. They get duplicated in interphase. So by the time it reaches prophase, you can see in this figure that the chromosomes are duplicated and they have 2 chromatids and joined at the centromere there. And in prophase, they condense and the nuclear envelope starts disappearing. Metaphase, the chromosomes line up in preparation for being pulled apart, and it's the spindle fibers that are pulling them to opposite sides. They are distributed evenly, and you can see in the end, after cytokinesis, we have 2 cells that have identical information, and they have all of the chromosomes of the original cell. They're not in the duplicated state yet, right? Not because they haven't entered interphase yet. So that's how body cells are made. In meiosis, the chromosomes do duplicate in interphase, but this is different. Prophase 1, the chromosomes are lined on top of each other. They have the ability then to cross over. So we have information, as eukaryotes, we have information from each of our parents. So that's why we have homologous pairs of chromosomes. And these pairs will line up and exchange, so giving an opportunity to mix it up a bit. So chromosome 1 in the end here is not going to be all from mother or all from father. It's going to have a little bit of each because of the crossing over. And then in metaphase, they lining up 2 by 2, not 1 by 1. So 2 by 2, they're lining up. And then in anaphase 1, we have it pulling apart, but the sisters do not separate. They're still, these chromosomes are still in their duplicated state. Here you can see that there was some crossing over. You can see some mixing of the colors. In telophase, they're now pushed to either end of the cell. And then we do have division, the cytokinesis. And then in meiosis 2, the chromosomes are condensing. If they have decondensed, they might not have. And then they'll line up in metaphase one by one at this point. And then the sister chromatids are pulled apart in telophase, anaphase. They're making their way to either end of the cell. I'm sorry, in telophase are making their way to either end of the cell membrane will return. There'll be cytokinesis event, and then we've got 4 cells. So it's 2 rounds of cell division. Only one time was the DNA duplicated. That was at the beginning. So that's why we go from diploid to haploid, and then we stay haploid. There's not another round of duplication of the DNA. Okay, next, let's talk about the concept of genome. Genome is all the genetic information that a cell might have. And for prokaryotes, we're talking about its bacterial chromosome, or it might be archaea is another type. So archaea and bacteria have 1 chromosome, and it's double-stranded. And they might also have plasmid or plasmids, which is also genetic material. It's outside of the main chromosome, and they're genes that aren't absolutely essential for the life of the bacteria archaea. The eukaryotic genome is a bit different in the sense that there are many linear, because they're not circular, they're called linear chromosomes. Here we're seeing one that's in the duplicated state. How many chromosomes depends on the species, but I can tell you one thing, it's going to be even numbered because they're in pairs. So we humans have 23 pairs or 46 chromosomes, and it's going to vary with the organism. And why pairs? Because our parents are in pairs too. And we received one from each parent. So we can see that again here with these karyotypes. A karyotype is, laying out the condensed images of the condensed chromosomes by chromosome number. And it's taken from a body cell. You know that because they're diploid. Body cells have diploid number of chromosomes. And now they're in pairs except for the sex chromosomes. The sex chromosomes, males have an X and a Y, females have 2 X's. So they aren't identical, as you can see, at least not for the males. And sometimes we get aneuploids, like you see here, and we can have an uneven number of chromosomes. Like for here, this is chromosome 21. We talked about Down syndrome. So they have 3 copies of 21, which is the cause for the symptoms of the Down syndrome. Now, if we're talking about gametes, there would only be 1 copy. So for humans, the normal number of chromosomes is 46. The number you can see here in the Down syndrome individual is 47, but normally it's 46. But in the gametes are half the number. And so whatever species it is, the gametes will be half that number. Like a pufferfish has 21 chromosomes in its egg or sperm. So it'll be half the number. So when it unites with another gamete, it'll bring it to the correct chromosome number. So it'll be these chromosomes and either X or Y for the gamete. Not many questions here. I guess you're saving them for later. But I am checking for the chat area. If you have a question, please just chime in. Talk about fertilization. It's an important part of this cell cycle because of the way eukaryotes are, where we produce gametes that have half the number of chromosomes. The parent, here, let's just look at this figure right here. This would be the total number of chromosomes is 6, just for this figure. Some species it has 6. So the parent this parent contributed 3 in its gametes. This parent contributed 3. You can see it as pairs. Now, what is going to be passed on to their gametes of this individual? It's going to be 3. Which 3? It's random. Any of these could be the case. And so it's random how in meiosis, how the chromosomes line up, and independent from each other chromosome, Mendel's law of segregation. And so the eggs that are produced by this woman are not going to be similar to the other eggs. They're all going to be individual. They're all going to be distinct. And the same with the male. His sperm, they're all going to be individual and distinct. And the union of those is going to be a combination that has never happened before. And so what that means is we get a unique individual that has never existed before, different combinations. And because this was haploid and this is haploid, their fusion, this haploid and this haploid, their fusion is going to make a diploid, zygote. And many different combinations are possible. And in addition to that, the egg and the sperm are a result of meiosis. And in meiosis, crossover events can happen. And so the combinations are even greater than that because of the possibility of crossover. So it gets chromosome 1 for mother, but what crossovers happened in the maternally derived chromosome, that there's many crossovers, many possibilities, many combinations, making for, like I said, unique egg and sperm. And so why all of this shuffling? Because variety is important. It's actually instrumental in evolution that we have different combinations that result. It makes it for individuals that might be better able to survive. So hopefully that was clear. Want to compare homozygous versus heterozygous. Homozygous means having 2 alleles that have the same information, like here or here. Or heterozygous means 2 alleles that have different information, like these 2. Genotype versus phenotype. So with respect to this particular gene, represented by capital A and small a, where capital A looks like it's for dark colored coloration in this moth. The genotype could be capital A, or capital A, small a, and there still would be the dark colored moth. So phenotype is really the physical manifestation of the observable traits is the phenotype, whereas genotype represents the genetic makeup. And so as you can see, they're not always the same. One that you can tell the genotype of is if you have an organism that has recessive traits, because the only way that it would show is if it had 2 copies of it. And so sometimes we can tell, but if for dominant traits like the dark color in moth, we can't just by looking. And so if we were to do a cross here, a monohybrid cross, that means we're just looking at the crossing one particular trait where they're different. These are the types of results that you get. I'm using peas, we talked about in class, but this example is the height of the pea plant. And we have a tall pea plant, and tall is dominant, and a short pea plant. And these are pure breeding. If you ever see pure breeding, that means that they're homozygous. So pure breeding means generation after generation. So we have pure breeding, tall, pure breeding, short. The only thing that can result from this cross, this is the P generation. The only thing you can get in the F1 generation is a capital T, small t, because that's all these parents have to give. And so if they're pure breeding. And if we then take this one step further, and the monohybrid crosses, it's very typical then to take the offspring of the F1 generation and cross it with itself or with a similar plant that was the result of this crossing, we will get well, this is a Punnett square, and then the Punnett square you should put the alleles, the possible alleles that might be in the eggs, might be in the sperm, and then it gives you statistic chances of what will appear in the offspring. And so with this tall plant and the tall F1 generation, we do see the return of the short plant because they were carriers carrying the small t. So it's possible the small t can unite with the small t, but that's only 25% of the time. So the phenotypic ratio is the physical appearance. And I like to create these Punnett squares. And you can do that on the back of your Scantron. You'll have a little space if you want to. Create the Punnett squares and then actually indicate what the plants are like. Then you would see this is tall, tall, tall. So 3 out of 4, or 3-to-1 ratio of tall to short. But the genotypic ratio is different than that. You see this is 3-to-1, this is 1 to 2-to-1. They're different because genotypic ratio is telling you the genetic components. 1 capital T, 1 small t, but there's 2 ways to make capital T, small t. So that's why it's a 1 to 2-to-1 ratio. But if you were just looking at the plants and you didn't have this prior information, you didn't this cross, and you have a tall plant, you might not know what its genotype is because you can't tell just by looking at the plant. So here's a tall pea plant, and maybe this is from the F2 generation. We're wondering what's its genotype. These are the 2 possibilities. It could be capital T, or it could be a capital T, small t. How does one tell? Well, you do a test cross. In a test cross, you cross it with one that you know the genotype for. And like I said, what we do know the genotypes for are one species that show the recessive trait, because the only way they can show that recessive trait is if they're recessive homozygotes for it. And so that's what's used in test crosses. You cross with a recessive homozygote. In this case, that's a short plant. Or crossing the unknown tall, unknown because we don't know the genotype, with the short plant. These are the 2 possibilities after the cross is performed. If it was indeed a heterozygote, capital T, small t, then we would expect half the offspring to be short. If it was dominant homozygote, capital T, we're putting that on the different parts of this Punnett square, then all of them are going to be tall. So that's one way you can tell. You can't tell by looking what its genes are, but if you're able to do a cross, you can deduce it. So let's talk more about the inheritance of traits. There's autosome, this means not the sex chromosome. So recessive traits, how are recessive traits passed? This is one possibility. We have 2 individuals that are heterozygotes, right? 2 different forms. And if these 2 heterozygotes, also called carriers, that means like carrying the trait, in this case, this mutation, but not actually showing it. If these 2 carriers are crossed, these 2 heterozygotes are crossed, they have a 1 in 4 chance of having an individual that has the trait in question here. So 1 in 4 individual that's affected. Let's continue just looking on this side of the slide for the moment. This affected child, it might be its albinism, for example. This affected individual, are they, must their children also be affected? That's my question to you all. Affected individual with an autosomal recessive trait, must their individuals be affected? What do you say? We got a 50-50 chance. The answer is no. Now I'm wondering, are you all there? Hello? I hope I'm not talking to myself here. Just say hi, somebody. Okay, thank you.
Yes, we're here.
So must their offspring be affected? And the answer actually is no. If this is a recessive trait, it's possible that this individual could mate with a homozygote that's unaffected. And if that person is not a carrier and they don't have this trait, then actually none of their children will be affected. And with recessive trait, the only way that they are going to show the trait is if they get 2 doses of it. So if this was a parent, it would pass on 1, but it still has to get a 2nd dose. So if it mates with the carrier, it might have an affected offspring, 50% chance, in fact. But if this person mates with someone who's unaffected, none of their children would show that trait because their children would receive a good copy of the gene from that parent. Autosomal dominant. Now, that is a different pattern of inheritance. This could be like dimples. This is an example we talked about in class. Autosomal dominant. Dwarfism is another one. In this case, the pattern is different, isn't it? It only need 1 copy for it to show. Here you needed 2 copies for it to show. You only need 1 copy for it to show. And here we've got a cross between an autosomal dominant person and someone who's unaffected. What their offspring look like, chances are half their offspring will receive this dominant trait because there's a 50-50 chance this individual is passing it on. And if he passes it on, it shows it's dominant, right? So that's why you have 50% of the offspring here that are affected because of his gametes, 50-50 chance. And she just passes on the normal version. Okay, moving on. X-linked has a different appearance to it. In X-linked traits, it's carried on the X chromosome. And so therefore, we can see with females, a female can be a carrier. A female can have 1 copy of it and it doesn't show. She carries it, but she's not showing, she's not affected. And whereas with males, if they have a copy of that mutation, it shows. There's no other X chromosome to mask it. So dominance and recessive, really, it doesn't really apply to males because they don't have the opportunity to have another chromosome masking it. So the pattern of inheritance here is he is going to be passing along his X, but when he passes on an X, that means he's creating daughters. His wife, who's unaffected, is passing on her X, so none of the children are going to be affected. Color blindness, hemophilia are examples of X-linked traits. However, here you got 2 individuals that are don't have the trait. One is carrying it, but they're not showing it. It's not their phenotype. But they can have an individual who is affected because when she passes her X on to any son, he's going to receive a Y from dad. That's what makes him a male, having X and Y. You can only get the Y from dad. And so the son will be affected. And so anyway, with X-linked recessive traits, you see it much more often in men because they don't have the opportunity to have a 2nd chromosome to cover it. It's always going to show. If they have it on their X, it will show. So let's compare incomplete dominance and codominance. In incomplete dominance, there is actually a blending. And so since it's not dominant recessive, we don't have capital and small letters, as you can see here. Both of them are given C, and then capital R subscript for capital W subscript for red or white. And so the F1 generation, when these true breeding plants cross, the only possibility is having a C capital R and a C capital W. So the only possibility is that it would be pink. Now, if you take this to the F2 generation, then you can see the return of these traits that we had of their grandparents, so to speak, can occur when you're crossing two heterozygotes. And the genotypic ratio here is 1 CR-CR to 2 CR-CW to 1 CW-CW. And the phenotypic ratio is 1 red to 2 pink to 1 white. So they're actually the same. Phenotypic and genotypic ratios are the same in incomplete dominance. Whereas codominance, it's not a blending like you see here, but both show, like in this roan cattle, when you cross this, I don't know, this brown color with this beige color, you don't have some in-between color that results. The brown appear as big splotches on the beige. And so we have the combinations of some strange noises. I just had to stop and listen. We have here the combination of the colors. It's not a blending. So that's what makes it codominance. So they both express. Here they're blending, here they're both expressed. But in the notation, neither of these do you have the capital and small letters. Polygenic inheritance is when you've got a number of genes all together, and they have alleles that each contribute some. So when there's a range, we've been talking a lot about, you know, very clear, distinctive, tall, short, round, wrinkled, etc. In polygenic inheritance, we have multiple genes and they each contribute some. This is a model that has skin color as 3 different genes, each contribute a bit, so you can have a range of phenotypes, not just a few. The other example of polygenic inheritance I've got for you here is in the Labrador, the labs. It's a contribution of 2 genes that are involved here that will control whether the dog is black or brown is one of the genes. And then the second set of genes represented by E is whether the dog is yellow. And as you can see, it overrides the brown. It doesn't matter what else this dog has, whether it has a big B, little b, 2 little b's, it does, big B, little b, it doesn't matter. If there's 2 E's, it's going to be coming out yellow. So polygenic in the sense that it's more than one gene. We've got this gene and this gene. So there's 2, poly meaning more than one. So in this case, it's two genes involved. And we use the word epistasis because the second gene here overrides, masks, whatever it might have on the first gene. All kinds of scientists we've talked about in these set of chapters. I'm going to talk about Erwin Chargaff. He's the one that figured out that the amount of adenine and thymine are equal and the amount of cytosine and guanine are equal. And he studied many different species, which was important for us in understanding the structure of DNA. Rosalind Franklin, she was involved in X-ray diffraction, and she found patterns that indicated that the DNA molecules are in a spiral shape. Taking those pieces of information further, Watson and Crick were able to work out the double-stranded helix and complementary base pairing of DNA. This is Carolus Linnaeus. He came up with binomial nomenclature, the two-name system for naming organisms. This is Charles Darwin, and then also Alfred Wallace. Both of them came up with the idea of natural selection. We commonly attribute it to Darwin, but actually both of them came up with it about the same time. And then these individuals are Peter and Rosemary Grant. I just figured I'd show pictures of them too. We talked about them because they were able to verify natural selection with beak sizes and the changing of beak sizes with weather conditions on the Galapagos Island. So they verified Charles Darwin's work on the Galapagos and natural selection and evolution. So DNA complementary base pairing is how it functions. And this shows you how they base pair. It has a lot to do with hydrogen bonds. Hydrogen bonds will help to keep the adenine with the thymine and the cytosine with the guanine, 2 or 3 hydrogen bonds. So they're not a terribly strong bond. They can separate, and that's important for a molecule that needs to unzip, to open up, as we see DNA does. And DNA is composed of sugar, phosphate, and nitrogenous base in the nucleotide. And so it's really just a series of nucleotide pairs that have the same compositions. So let's compare replication to protein synthesis. So replication happens prior to cell division. It happens in the nucleus. We saw that it happens in the S phase and interphase. And so how it happens is that DNA has to open, like I said, and it does with the help of an enzyme called helicase. And then what that does is it exposes the nucleotides, and at this replication fork, we have 3 nucleotides that are available, joining with the nucleotides in a complementary fashion. Where there's a T, an adenine will come. Where there's a cytosine, guanine will come. And they are added to not just one strand, but both strands. So that's different. We're going to see that transcription requires or uses one strand. With replication, it's both strands that nucleotides are added to. And as we go and add nucleotides, we're going to end up with 2 identical, hopefully there's no mutations, 2 identical strands. And each of the new strands is going to have one of the original strands, and then one that's new nucleotides added to it. So one original and one with new nucleotides. And because they get one original and one new, that type of replication is called semi-conservative. Conserves semi. It keeps one and gets a new one. So comparing this to protein synthesis, the other important thing that DNA is involved in protein synthesis is a 2-step. There's the transcription process, and then there's the translation process, which, by the way, are in alphabetical order, if you ever get confused. And in transcription, which happens in the nucleus, we have transcription factors that will attach onto the DNA. And then RNA polymerase is an enzyme. It's not shown here, but there's an enzyme called RNA polymerase that will come and add. Now, how does it add? It's adding RNA nucleotides. So wherever there's a cytosine, it's going to put the RNA nucleotide guanine. Wherever there is an adenine, it's going to put the RNA nucleotide uracil. There's no thymine in RNA. And so anyway, it's going to add the A's, C's, G's, and U's, and RNA nucleotides. And then that'll come off. And before it goes to translation, there's actually parts that are removed. The introns are removed. The exons are then assembled together, which is strange to make something and immediately take it out, but there you have it. So the exons then will join, and then that leaves the nucleus. Now we've got translation, and that is converting this RNA information into protein, which is the point, of course, right? Protein synthesis. And so that involves tRNAs and adding tRNAs that will bind. I'm going to turn the speaker down. I'm not sure if you get some background noise, but I'm going to go ahead and mute that. Maybe if you know it's you, you can mute your microphone. Okay, great. We're back. Okay. So in translation, what happens is that the codons in the messenger RNA are going to combine with anticodons found on these tRNAs. It's going to be a lot of tRNAs available for this, and the tRNAs are carrying amino acids on them. And they're going to bring the correct amino acid, the one that the code is calling for. There's a codon, a 3-letter codon, will come because the anticodon, which is complementary to the codon, will ensure that the correct amino acid is coming into place. And then that continues and on until finally reach a stop codon, and that would then indicate that this process is done and the protein is assembled. And so you will have be asked to transcribe and translate, and I will give you this genetic table. So let's just take a look at this one, for example. If this is a DNA, double-stranded DNA molecule, and to transcribe it, we need to know which strand needs to be transcribed. So the template strand here is marked, the strand to be transcribed. So now we know. And that's to transcribe it, wherever there's a T, you're going to put an A RNA nucleotide. Wherever there's an A, you're going to put a U. Remember, there's no thymine RNA nucleotides. And then you continue on in that fashion. Wherever there's an A, you put a U. Wherever there's a T, you put an A. Wherever there's a C, you put a G. And then this can then be translated using this table. And so you just look up in the table for each of the codons and the messenger RNA. You can think of this table as a messenger RNA translation table. So if you look up AUG, you see it's MET, which is codes for methionine, AAG, which is lysine, UUU, phenylalanine. But if you look up UAG here, you see that it doesn't code for anything. That's actually stop. And so that means that's the end of the protein. And this shows you how this then is assembled, where you have the, if this is AUG, we've got or maybe we'll do one UUU. With UUU, we have a tRNA that has in its anticodon AAA, and that tRNA is carrying phenylalanine. Yes, I'm going to make a recording of this. No problem. And no, I wouldn't have you memorize the mRNA table. I'll provide that to you. Thank you for your questions. I was beginning to wonder if anyone was out there. Okay, so just be able to transcribe and translate. And there's a couple, there's one that's assigned to you in the study guide, and then there's one that's in that problem too you can practice with. Okay, so I wanted to talk about DNA technologies and compare some of these very important processes. This is a big one, gel electrophoresis, because that was the first way we were able to separate DNA fragments. DNA can be cut, as you know, with restriction enzymes, but it can be drawn apart. And the way that you put it into this gel, and then you run electric current through it. And because DNA is negatively charged, then it's going to travel towards the positive cathode, and little pieces will be able to make their way easier. So little pieces are going to travel further, larger pieces not as far. So we can separate pieces of cut DNA. And that has been of great benefit because it gives distinctive patterns for one thing. And so my DNA and your DNA, we'd have different looking patterns because we have different codes, so they'd be cut at different places by these enzymes. So it can be used for DNA fingerprinting. It can be used for identifying parents for paternal. It can be used for identifying diseases. So, because each organism has its own distinctive DNA fingerprint, which is kind of like a scan code because of the pieces and the separation of the pieces. This is PCR, and that is a technology, polymerase chain reaction, where segments of DNA can be increased dramatically in their number. And what that happens, that's done using cycles of heating. So it's all programmed. So it heats and has the presence of enzymes that can copy. So when it heats, the DNA strands come apart because that breaks up the hydrogen bonds. And then primers are added. You'd have to know something about the sequence that you're copying. So you'd add a primer that would pair to one end of that sequence, and then adding nucleotides in the presence of DNA polymerase. And they use a DNA polymerase that can operate at high temperatures. So basically we're copying. It's what we're doing is we're doing DNA replication. We're providing all the pieces for DNA replication. And then with the cycling cooling off, it would then pair, the two strands would pair, and then that can be repeated and repeated and repeated. And since you increase from 1 to 2 to 4 to 8 to 16, it gets logarithmic, and you can get rapidly large numbers of copies. So basically it's for copying pieces of DNA, which are needed for a lot of these technologies. You need not just one piece of DNA, you need it in multiple copies. DNA sequencing, that's to determine the actual sequence of nucleotides in a fragment of DNA or in a whole genome. So it's for determining nucleotide sequences, but you do need to fragment into pieces to do this. If you're going to do a genome, then you put the pieces together like a puzzle. But anyway, you fragment the DNA into small pieces, and then they are copied, and then oh, excuse me, I'm looking at the wrong thing. So what you're going to do is then you're going to provide for creating a copy of the other strand. You're going to provide the A's, T's, C's, and G's, but you're going to provide an altered copy of some of the nucleotides. So they'll have fluorescent tags, and that they're terminator bases. And so when the DNA fragments are paired with nucleotides, it goes along just fine until one of the terminator sequences come in, which is just a small fraction of the nucleotides you're providing. And that terminator sequence also has a fluorescent color tag. So in the end, not that you need to know this level of detail, but in the end, what you'll have is a series of fragments of different lengths, and they will have a terminator on their last base. And these can then be passed through a gel, somewhat like the gel electrophoresis, based on size, and the fluorescence can be detected because they'll light up a different color when laser light is passed through them. And what you can find out in the end is of these fragments of increasing size, what's the last base pair? And then you can from that come up with the DNA sequence. And so anyway, this is a great technique for figuring out the code of life, the A's, T's, C's, and G's. And so they've been able to sequence all kinds of genomes, including the human genome. Now, CRISPR, which is what you have your discussion on this week, it's an RNA-guided enzyme that can take the enzyme directly to a place in a chromosome. Because of the RNA guide, it's looking for a specific place. It's kind of like a GPS in a way. So it takes the enzyme to a certain location on a chromosome where it then snips it. And so this can then interrupt a gene, or another piece of DNA can be inserted into that spot. CRISPR is an amazing technology because of its accuracy, and you can direct it, having it go to a specific location, and make the change in a very accurate way. So it's like programming. It's like programming a chromosome by putting another piece exactly where you would like it. And from that, they've been able to create drought-tolerant plants or hardier plants. They've been able to edit human genome. Those are the types of experiments that are happening right now, actually, in China. It's possible that you can also correct disease-causing genes to basically put a normal copy instead of an affected copy, and to overcome a disease. So they did sequence the human genome, and it's interesting what they found when they sequenced the human genome is that a lot of it was really repetitive. They were sequences that were repeated over and over and over, like ATTG, for example, where it's kind of like a stuttering in a way. Much of the genome really doesn't even code for proteins. That was the biggest surprise. Only about 1.5% of genetic material codes for proteins, RNAs and tRNAs. The rest of it is either introns. We talked about introns being removed. Introns, a significant part, but the bulk of it are transposons, or transposable elements, which are repetitive sequences that can insert themselves in different places in chromosomes. And so they're repetitive, and they don't, as far as we can tell, they don't serve the organism, but nevertheless, we carry them. Eukaryotes, not just humans. Eukaryotes carry transposons. That's the bulk of it. Okay, I wanted to now compare molecular cloning with reproductive cloning. So molecular cloning requires restriction endonucleases. And what happens is that you cut some foreign DNA, it can be any source, and very commonly DNA of a bacterial plasmid, because they're so convenient for this. And, the restriction enzymes will cut in a staggered way. And so you're able to then insert two sources, in this case, a bacterial DNA with, we'll say perhaps a human, maybe it's human insulin gene. And then this two-sourced plasmid now can then be inserted into bacteria, and the bacteria just read it as if it was their code and produce the protein. And so in this way, they've been able to create products like insulin and human growth hormone, etc. Molecular cloning has been done also with plants. They've been able to insert into, I don't have a picture of it, but they've been able to insert into plant cells particular genes of interest that have led to resistance to disease, resistance to herbicide, like for example, Roundup. So molecular cloning, we're taking a molecule and inserting it, a molecule of DNA, and inserting it into an organism. And then the result is going to be a genetically modified organism. Whereas reproductive cloning, and there's two outcomes here I'm going to talk about, is where you take, and the genetic information is the whole nucleus. Here it was just a gene or molecules that were coming out. Here we're going to take the whole nucleus. In this case, of this Finn Dorset sheep. They took some body cells, happen to be mammary cells, and the nucleus of the mammary cell, as you can see, is here, and it's going to be joined with the enucleated egg. So this sheep is providing this egg cell, but its nucleus has been taken out, enucleated. And so all the genetic information is from one source, and as opposed to this one is mixing. So it's all from one source, and then they found that they were able to get it to merge with a direct current pulse and create a clone. And what we're cloning here is the organism. So the outcome here doesn't really matter who carries it. It can be a surrogate, because the mother is not even contributing. I guess mother is so confusing a term. This surrogate you is not contributing any of its genetic information, but it is carrying this embryo, this embryo, and the dolly was the first one cloned sheep that was done in this way, would be identical to the nuclear donor, because that's the only genetic information this organism had. So that's why they call it reproductive cloning. Now, reproductive cloning is a useful way that people have been using to get stem cells. And a very similar situation here. We've got a nucleus-free or a nucleated egg. And so this woman is donating the egg. But another organism, another person has been the source of the nucleus that was added to it. And so all from one source. And the reason someone might want to do this is because this fertilized egg will then develop and develop into the blastula stage. And in the blastula stage, cells are not defined on what part of the body they will become yet. So they're totipotent. And so they can become any type of cell, in fact. And so having stem cells that can become any type of cell has a lot of promise to help recovery of people that might have damaged body parts, or so it's possible that you could have stem cells, your stem cells, because if the source of the nucleus was you could have your stem cells produced, and then that could be used to make up any body cell. And so this is reproductive cloning, but it's reproductive cloning, basically making it to the blastocyst stage. But that's the goal, is just to get to the blastocyst stage, to get the stem cells that you get in the blastocyst stage. It's not that the embryo is now going to be in a surrogate. That is not, as far as I know, that's not done. We don't have any human cloning. Okay, moving along here. Thank you for sticking with me. I keep checking on the chat to see if you have any questions. I'm going to talk about evolution right now, and the process of natural selection and its outcome. So with this very simple diagram, we can say that mutation is a random event. It's really errors in DNA, copying DNA, or it could be from exposure to mutagens, but it's random on how it affects the genome. And chances are it's not going to affect the genome in a way that is going to be helpful. But every once in a while, it can be helpful. And so random mutations here represented here with lots of different shapes that result. And then, however, the process of natural selection is not random. It is, it has to do with the fact that there's typically less resources than are possible for all offspring to survive. So it's survival of the fittest in a way. And in that way, whichever of these mutations would allow for better survival, that is going to be represented more and more in the offspring. Let's take a very a simple example here of the tortoises in the Galapagos. And so in these larger islands where there's plenty of grass, this is the type of turtle body that you're going to see there, nice domed shell. However, on the smaller islands like Hood Island, where the grassland is very minimal, there's cactus, the tortoises there have more of a saddleback, and they're able to reach up and eat the cactus. And so this resulted from a mutation. And the mutation that happened to be helpful in this case was for a shell that allowed the tortoise to be able to stretch his neck higher and reach that. So the first event here, that is the mutation to a different shaped shell, was random. However, of all the tortoises that were born here, the ones that could eat better, that is reach the cactus, are going to produce more offspring. And so that's what I mean by non-random. Natural selection is a non-random process. And so the frequency in the population of these traits is going to be higher and higher. And it's going to result in organisms that are better adapted to live in their environment. And so that's how all of our traits that are particularly well suited for our life and other organisms' traits, that's how they came about. Originally from random mutation and then being selected. Okay, study guide, question number 7. What are the underlying causes? Let's see. Pull that up here. So the underlying causes, it's a good time to ask for the question. Thank you, Diana. Is the random mutations, and then descent with modification. So that is passing it, the random mutation is in the genes now. So descent with modification talks about how they are then pass them onto their offspring and their offspring. And then natural selection. And natural selection is the non-random process where organisms that are better able to compete for the limited resources are going to survive better. And they're going to produce more offspring. And these offspring will be more suited to live in their particular environment. And then added up over time, you're going to see more and more of those traits. Another example of that would be, like I was talking about, the grants, looking at bird beaks in the Galapagos Islands. As in the dry years, having a harder, stronger beak would allow them to survive better. It was random, the change that led to the beak changing. That was mutation. But it is not random, the fact that they're able to survive better, and so more and more of them. Okay, so mutation, yes. Genetic flow, genetic drift, natural selection. Flow and drift might occur, but they're not essential. Underlying causes. I'm thinking mutation and natural selection. That's what I'm looking for, but I'm not saying that you're wrong, because those are perfectly good points to talk about too, genetic flow and genetic drift. But essential would be mutation and natural selection. Thanks for helping each other out. And moving on. Okay, so this slide is actually my last slide. And this one is about the lines of evidence supporting evolution. So evolution is not controversial among scientists because there are many lines of evidence to support it. And one of them being the fossil line of evidence. And in the fossil line of evidence, you see a slow progression in the fossils record, one organism to another, to another, to another. This happens to be, oh, actually it's this way. This happens to be the fossil history of the whale. Something interesting about the fossil history of the whale is the fact that it evolved from a four-legged creature. And what is still in some whales today is the remnant of a hip bone. And obviously a whale would not have any function for a hip bone, but it's vestigial, meaning that it's part of a remnant of anatomy of its ancestors that no longer has any use. And that helps to support the idea that it had an evolutionary past. Biogeography, it's when the best explanation for the distribution of organisms is the movement of tectonic plates. Remember the big picture here. These countries were at one time adjacent to one another, and they all carry this type of plant, this protea flower. But once separated, then they each went their own separate way, and so their own evolutionary path. And so obviously they're related, but the ones that you find here are different than the ones here, different than the ones here. So the easiest explanation for that is that they had a common ancestor and then different evolutionary paths leading to unique regional species. The molecular biology evidence is the universality of the genetic code. All living things, I'll get to your questions in just a second. I do see them there. That all living things have the share genetic code. And in fact, organisms that are more related to each other share more of that code than organisms that are more distantly related to each other. And that's what you would expect if mutations are accumulating over time, that there would be more and more differences as organisms are less and less related to one another. Anatomical evidence, like for example, the arm, leg bone, wing, and different organisms have a similar pattern in how they're put together. And the best way to explain that, the similarity in the overall layout of these appendages is that there was a common ancestor, and these are all descendants from a common ancestor. Similarly, with this embryological evidence, that there's similarities, actually it's hard to distinguish these, right, in the embryos of vertebrates. And the fact that there is this similarity indicates a common ancestor and shared ancestry. And the different stages are kind of remnants of the shared path that we have with these organisms.